Hello all, welcome back to the BioRocks channel. Today in this video, I'm going to teach you about the structure of the myofibril. So the topic that we are learning today is going to be the structure of myofibril. You remember children, I have introduced you saying you that when you're going to cut a muscle, this is a muscle belly that you're seeing. We spoke that they are going to have fascicles. Fascicles are going to have, so this is your fascicle, right? Fascicles are going to have muscle fibers. Then what we could see was if this was a fascicle and this was a muscle fiber, inside the muscle fiber, there was another minute structure which was being called as myofibril, right? We have seen about this, and I even made a special video regarding only the structure of the myofibril, where I spoke that the sarcoplasm, the cytoplasm of this muscle fiber, is going to have these myofibrils, due to which they are going to get developed some striations, right? So those we are going to see in detail. I bet you that none other video might have made this whole structure of myofibril, and I bet you that this video is one of its kind, which is going to make your subject more clear by giving you the concepts of further need. So do please subscribe my channel and try to share my channel with others. Do hit like the like button, as well as please do comment in the comment section how you have liked this video. So what I'll do is I'll start up this in detail, and I bet you that this is going to actually make your structure of muscle and the structure of myofibril the most interesting, the most easiest, such that you will score all the marks of the questions that will be posed in the boards, as well as a need from this particular topic. So children, that you're seeing on the screen now is the structure of the myofibril. So this particular thing that you're seeing in the red color on the screen is going to be actually the thick filament. Okay, children. So this is the thick filament. And you can see that this whole length of the thick filament, since it's darker in color, we call this as an isotropic band. We call this as an isotropic, or we call in short as A band. Since this is going to look darker, we call it as an isotropic band, but this is actually a thick filament. And you can even see on the thick filament that there are some structures like this, which are being called as a myosin. This is also protein. You remember, children, when I made a structure of muscle, I spoke that myofibrils is made up of thousands of proteins, right? So today, whichever we are seeing here, the whole structure that you're seeing now here is going to be a myofibril, as I told you. So this is an anisotropic band. So all the red ones, this and this, as well as these, whatever you're seeing on the screen in red color, the darker ones are being called as anisotropic bands. Actually, they are the thick filaments. And you can see that there are myosins. You can see these green colored structures here. These are going to be the myosins which are arising from this particular thick filament, okay? And you can even see that there is a blue protein, right, children? This particular protein that you are seeing inside, this particular protein that you're seeing inside a thick filament or A band is going to be called as a titin protein. This is called as titin protein. You can see that the titin protein is even extending outside. This is also a titin protein. And what is this titin protein making is, this is trying to fix your thick filament with the Z disc here. Z disc, okay? So its whole function is to actually try to what you call is stabilize the thick filament, okay? It is stabilizing the thick filament to the titin protein which is inside. So this is a titin protein which is running outside and fixing your A band to that of this your thick filament to the Z disc. So that you are seeing here now on your screen, which is this particular structure which I have drew. This is a Z disc or Z line that you're going to call as this whole structure in the blue, which is running all the length in between the thin filaments, is going to be the Z disc. So let me try to make the Z disc here also. So you can see this one that I am making here, the blue, the black and red, this whole structure is going to be called as the Z disc, or you can even call it as Z line we are going to call. So this is your Z disc. So we have seen a thick filament, red colored ones. We have seen a blue protein inside that, which has been called as titin protein. So here also you can see the titin protein present here. And these two long ones which are running here are being called as a Z discs. Actually, the length or the distance between the two of the Z discs is being called as a sarcomere. So this whole thing that I have drew here in red color, the big line here, is going to be the sarcomere. So what is a sarcomere is the distance between the two of the Z discs, are the Z lines, are being called as sarcomere. And sarcomere is actually a functional unit of your muscle fibril, that is a myofibril, because you can see the thin filaments in it. Let me introduce you those and then I'll cover that particular topic. So children, you have seen the red ones, which are darker in color, the thick filaments having a protein called as titin protein, which is fixing the thick filament with the Z disc here, right? Then you could see the myosins that are arising, that you can see here. And next thing that I would like to introduce you is this whole blue bead-like structures here, this whole length structure, which is running in between of the thick filament, is being called as a M line. So this is being called as M line. Who is M line? These bead-like structures are being called as a M line, we are going to call. So what is this M line made of? M line is made up of actually what we call as some of these proteins, okay children. And what are those proteins? We will see. Let me write these proteins for you such that it will be easy for you all children to understand this structure far better. So the thing that I'm speaking is about the proteins present inside the M line. So the proteins which are making these are the first thing, creatine, creatine phosphate. Creatine phosphate is one of the protein. And another protein which is making the M line is going to be called as myomycin. This is also a protein. Okay, myomycin, creatine phosphate, as well as you can see that there is another protein which is being called as a C protein. We call this as C protein. So your M line is made up of three proteins. It is made up of how many? Three proteins. And what are those proteins? The C protein, the creatine phosphate, myomycin are the proteins which is making your M line. And this M line is running all through the middle of this particular red filament or the thick filament over here. Okay, so that is M line there. Next thing is that you can see here, observe here, is the distance between the two thin filaments. So before that, I'll give you what is thin filament. So the whole structure that you are seeing from this portion to this portion, along with all these bead-like structures, is going to be the thin filament which is being present. Okay. So this black structure which is running in between is being called as a nebulin protein. It is a nebulin protein. And all these proteins here, the globular bead-like structures here, are being called as your actin protein. Actin protein. So your thin filaments are made of a protein according to NCRT as actin protein. So that is going to be your thin filament. But who is holding the thin filaments in between is going to be the nebulin protein, right children? That is going to be the nebulin protein. And next thing that you can see here is the distance between two of these A bands from here, okay, to that of here, this whole distance, okay, that is being called as a I band, we are going to call. Why do we call this distance between the two darker bands, that is your thick filaments? This distance here is being called as I band. I stands for isotropic band because it is light in color, light. What are all present here in the isotropic band? See, you're having these actins, you're having the Z disc, you're having the titin proteins, all these. So the thin filaments are there, the Z disc is there. So whole together, this is being called as I band. So you can see this is a dark band, right? This is a light band, this is a dark band, this is a light band, this is a dark band. So every time a dark and a light, a dark and a light, and a dark and a light. That is the reason we can see the presence of striations in the skeletal muscle. Okay, the skeletal muscle has got its striations just because of the presence of this thick as well as that is isotropic as well as anisotropic bands, children. I hope I made it clear now. Okay. But what is that you are able to see here? The green color structure that I drew here, and a structure like this, which is blue in color, and what are all these? We will learn it in detail. Let me take a separate slide further and explain you what are these structures. If you understand those three structures, then it will be easy for you all to remember it in a very good manner. So first what I'll do is I'll go with the beads now. Okay, so what I'll do is I'll choose a color for that and try to draw those beads. So these are the beads, children, that you're seeing, which I spoke that the thin filament is going to contain a protein called as actin, right? So these are the actin proteins, and actin proteins are going to have what? They're going to have a, I drew like this, then these are none other than the active sites where the myosin heads are going to be attached. So let me label this for you. This whole circular structure is being called as a actin, but this actin is called as a globular actin. So we call it as G actin. Okay, so such G actins, they act as monomers. They all togetherly polymerize. What is polymerize? The repetitive units. These monomers are going to repetitively join together. Okay, so the G actin is getting polymerized, right? So the G actin is getting polymerized to form what? A filamentous actin. It is forming a filamentous actin, which we are going to call as F-actin. So what is present here is F-actin. What is this whole structure, children? This is none other than your thin filament, right? This is your thin filament. Okay, we are seeing the structure. Let me show you where is that on the screen. So this is what I'm telling. This is what I'm telling. So these are your actins, globular actins togetherly forming the polymerizing to form a filamentous actin. And can you see here, I made some black colored dots over the actin. Those are going to be your ones which are being called as the active sites. So this is going to be the active site. Actually, children, you can see that from the thick band, there are some structures like this. I told you that they're being called as what? They're being called as myosin. So myosin attaches to these particular active sites of the thin filament. So here, this is the site where the myosin binds. Myosin binds during what? During the muscle contraction. So during the muscle contraction, they are going to bind with each other. Okay, that is the one. So this is what you are seeing is going to be the one. Now children, if you are going to observe what I have drew here, I drew a green color structure running on these particular actin active sites, right? Covering it up completely, this green structure. So what is that called as? So that is none other than your what we call as the tropomyosin. Tropomyosin is going to be like a layer which is covering up your active sites of the actin protein. So this is covering up. So that is being called as what? That is called as your tropomyosin. So tropomyosin, what it is trying to do, it is covering the active sites. Active sites of whom? Active sites of the actin protein. This is seen during which process? During the relaxation. During relaxation, the active sites of the actin are covered by tropomyosin. Okay, this is the one. Then what did we see here? We can see that I drew some structures like this, right? These structures are being called, see here also you can see this structure here. So what are these structures called as? They are being called as troponin. Okay, troponin. So what the troponin does and what is the structure of troponin? Let me write it here. I'm taking a troponin now. So if you look at a troponin, it's going to be a protein this way. Okay, this site where the calcium comes and sits here. Okay, it is going to be the calcium binding site. So this is going to be your calcium. Let me write this for you all. So this will be calcium binding site. So we call this site of troponin as troponin C. Okay, now where is the troponin? Troponin is this way. Okay, so it pulls away. When the calcium comes, it pulls away this particular tropomyosin. So one is a calcium site, troponin C site, and one site is going to be actually tropomyosin binding site. Right? I told you that it is going to pull away this green color structure. So that is one end is attaching to the tropomyosin. So this is going to be the tropomyosin binding site. Okay, so we call this as troponin T. Now, children, the other one is going to be the other arm is actin binding site. So one is attaching to the actin, as you can see here. So actin binding site. So what is this called as? This is called as troponin I. Okay, this is called as troponin I site. This is called as troponin C site. This is called as troponin T site. Okay, this is actually a inhibitory site. I stands for inhibitory. Okay, children, that is troponin. What else is being left over? Tell me. Then we need to see about the myosin now. Where is myosin? I'll show you where is the myosin. So this is your myosin which is being present. It has two heads like this. So let me show you the structure first. So you can see that arising from this particular thick filament, you are having these structures, right? They are being called as myosin as I labeled it here. So what is that myosin is doing, children? The structure we'll see first. So as you see, this whole structure, let me take another color, it will be called as a tail. Okay, and this is being called as a neck. And these heads, these are the two heads. And in the head, you can see that there is a specific site here, and this is being called as ATPase. Here is the ATPase. Okay, so this has been called as myosin ATPase site. So ATPase, what it does is, this is the head. These are these sites which are going to actually bind to the active sites. Okay, so I'll write it here for you. The head binds with whom? With the active site of actin. This is the one. And children, in the tail structure, you can see two proteins here, which are the regulatory proteins, which are being called as light chains and a heavy chain. Okay, it's been called as a light as well as a heavy chain, which are going to regulate the movement of the myosin heads well. So this is the filamentous actin, this is a troponin structure, these are the myosin structures. So as you can see, we covered up the thick filaments here, and we could see the myosins. Okay, so we could even see the M line, we could see this. So now what I'll do is, I'll not go in this detail, but I'll draw a simple diagram for you children, such that it will be easy for you all to understand this. So as you can see, simply in a simple terms, this is Z disc, right? This is a Z disc. Now children, we took what? We took a thick band, right? So let me draw the thick band for you. So this is your thick filament. Thick filament. Okay. Next, what did we see? We could see that there is a thin filament, right? Going on here. Okay. Thin filament. This is your thin filament. This is your thin filament made up of filamentous actin. And these are. And next, you could see that from the what do you call these ones. What are those? From the thick filament, you could see that the myosins are arising like this. So these are the myosins that you can see. On the actin, children, you could even see some other structure, right? And that structure was like covering up the active sites, and that was being called as tropomyosin. So this was covering the active site. So this is like a layer of protein which is covering it up. So let me write, this is going to be the tropomyosin, right? And the one which is running in between is going to be the Z line or Z disc. And this whole structure is going to be thin filament. And the thin filament is being made up of actin proteins. And you could see then the what you call is a thick filament here. This is your thick filament, children. And next we could see was this whole structure was being called as a myosin. So this is your myosin. So myosin heads here. So, and you could even see another structure like this, right? One is for the actin protein, one for the tropomyosin. What was this called as? This was being called as troponin, right? This was troponin. So what we need to know more else is going to be here. From the thick filament, there was a pre-protein structure here, which was being called as what? As M line. And we could see the Z line, M line, and the distance between the two of the Z discs here, from here to the here. So this is also Z line, right? So this was being called as what? The whole length was being called as a sarcomere. The fundamental functional unit of the myofibril here, okay, the muscle sarcomere. Now the what else we had seen? Yeah, now the distance between the two of the thin filaments, the distance between the two lengths of the thin filaments is being called as your H zone, we are going to call, okay. And what else, children? We have seen an M line here. So M line, you came to know the distance between the two Z lines is a sarcomere. We have seen the H zone, and this whole length of the thick filament will be called as what I told you, A band, the dark band. And children, what else is there? The whole length of this particular thin filament will be called as what? I band, the lighter band. So next again, you will have a thick one. So the things that you need to understand here is H zone is going to be what? So let me write it in a side for you such that it will be easy for you to revise when you're going to go for exams. So what is H zone, children? H zone is going to be distance between the distance between two adjacent two adjacent thin filaments, right? That is a one. Next, what is sarcomere? Sarcomere is going to be the distance between two adjacent Z lines, right? So that is a one. Next, what is a H zone you have seen, and what is the A band? A band is the whole length of thick filament, right? Whole length of thick filament, thick filament. Then what is I band, children? I band is going to be the whole length of thin filament, whole length of the thin filament. So these are the things that you need to understand. Okay, so these are the thick filaments, the thin filaments, and we are having these things like tropomyosin, troponin. These are the detailed structures of the what we call as a myofibril structure. I hope, children, I made it clear for you through this particular video, the actual concept of what are the proteins. What are the proteins we have seen? The titin proteins, the myosin proteins, the C proteins, the creatine phosphates, the myomesins, the actin protein, the nebulin proteins. So these are all the different types of proteins that you can see. Okay. In the next coming video, what I'll do is I'll try to show you the concept of the sarcomere during the contractions and relaxations. Let me show you a slide of this such that it will be easy for you all to even correlate it with the images of your textbook, what I have taught you now, with that of your textbooks, children. So let me show you this. Yeah, I have this slide for you all. So you can see that this is a Z disk running in between. So the distance between the two of the Z disks is being called as a sarcomere, we are going to call. And this is a thick filament, okay, thick filament, the A band, the whole length of the thick filament is called as A band, having the myosin heads here. These are the myosin heads, okay. And what else you can see here? You can see that the in between of these thick band, you can see a M line passing made up of three proteins, right? And you can see that the thin filaments are made up of this globular actin, which polymerize to form a filamentous actin. You can see the green colored layer covering the active sites called as tropomyosin. You can see these blue structures which are being called as a troponin, okay. And what is, children? What is the H zone? H zone is a distance between the two of the thin filaments here. Okay, you can see the titin protein here. The green color structure here he has given you is a titin protein. And actually the filamentous actin is going to run on the nebulin protein also. So this is a detailed structure of the myofibril. I request you all to do please share this video to many of the children, because this is a one which is not clear for many. So if they go through and if they are well versed with this information, then it becomes easy for them to appear for the examinations to solve the questions from this particular topic. I hope you all have understood, children. And in the next forthcoming videos, I'm going to make about the contractions of the sarco, that is a contraction of the muscle. We will see what are all the changes that are going to happen in the unit of the sarcomere. If you like the video, please do hit the like button and please do comment in the comment section. That is going to boost us. Thank you all. Thank you.